Hello everyone, my name is Antonio and I am postdoctoral fellow here at Stanford. Today I'll be presenting on the role of MAPK kinase ERK signaling at the crossroad between regeneration and fibrosis during skin and cartilage repair. The injury response of most human organs following a deep skin wound, myocardial infarction, stroke, spinal cord injury or lung infection heals with scarring. This pathological healing outcome consists in the replacement of the normal parenchyma with fibrotic tissue, defined as tissue hardening and the excessive deposition of collagens that compromise the tissue function. Among mammals, spiny mice have evolved outstanding regenerative abilities, able to regenerate complex tissue structures such as full-thickness skin, hair follicles, adipose tissue, nerves, skeletal muscles, and elastic cartilage. Spiny mice also exhibit enhanced functional recovery upon myocardial infarction, kidney and spinal cord injury, while laboratory mice heal injuries with scarring. However, the molecular mechanisms underlying these diverging healing outcomes are poorly understood. This study's ultimate aim consists of exploring whether adult mammals have completely lost their ability to regenerate or they still possess related regenerative traits. If the latter hypothesis holds true, it would imply that a regenerative response can still be triggered in poorly regenerative mammals, and it would have profound implications for regenerative medicine. Being ERK activated upon injury, from planarians to zebrafish and axolotls, it prospects as an ideal candidate to understand key differences between regenerators and poor regenerators among mammals. To test the hypothesis that ERK activity might differentially regulate the healing outcome, I employed a 4 mm ER punch as wound model and assessed potential differences in the ERK regulation between regenerators and poor regenerators. I asked whether ERK activation was functionally required for spiny mice regeneration to occur through loss-of-function experiments, and I performed bulk and single-cell RNA profiling to define the ERK-responsive cell types and downstream targets activated during regeneration. Finally, by using gain-of-function experiments to in vivo delivery of ERK activators, I aimed at stimulating a regenerative response in poor regenerators. I found that while ERK is immediately induced upon injury in both species, its activation is sustained over time only during regeneration. And phospho-ERK positive cells, labeled in green, populated the accumis blastema, the new tissue, in the epidermis and dermis. Next, we asked whether ERK had a role in spiny mice regeneration. After confirming that the treatment with the phospho-ERK inhibitor indicated as PD was effective and the ERK signal was gone by Western blot and immunohistochemistry, we pharmacologically inhibited ERK in the early phase of injury response and demonstrated that the wound sealing was completely impaired, as you can see by histology with the two epidermal margins unable to close the wound. When we inhibited ERK after revitalization occurred, we found that regeneration per se was impaired. To better understand the role of ERK, we performed RNA profiling on punched ears collected at 15 days after ear punch with or without PD treatment for only 5 days, and found that the top differentially expressed genes fell in these categories that are also the hallmarks of blastema formation, and I will lead you to some of them. Starting from the cell cycle, the number of EDU incorporating cells and mitotic cells decreased upon ERK inhibition. As for the formation of newer follicles in the wound epidermis, the number of LEF1 positive cells, a marker of hair placodes, drastically decreased upon ERK inhibition, suggesting that de novo hair follicle formation is ERK dependent. Regarding the extracellular matrix, we found that upon ERK inhibition, MMP9 expression was decreased at protein and RNA level, and the abundance of collagen was increased, resembling the fibrotic wound resolution observed in laboratory mice. The TGF-beta receptors were also upregulated upon ERK inhibition, and many studies show that TGF-beta signaling is a major player of most organ fibrosis. This data suggests that the ERK inhibition shifts the accumis regeneration towards a fibrotic healing outcome typically of laboratory mice. When we inhibited FGF receptor and RB2 receptor, the ER closure was compromised and ERK activity was drastically reduced, indicating that FGF receptor and RBB2 act as upstream ERK regulators. The next step was to see if we could stimulate regeneration in scarring wounds. I implanted microbeads soaked with growth factors in the punched ears of lab mice and found that not only the cell density was much higher, but newer follicles were formed in the new tissue, and the ERK closure was increased compared to the control group. The new tissue was enriched of phospho-ERK positive cells and proliferating cells. In addition, the number of alpha-SMA positive stromal cells associated with fibrosis was significantly decreased in the treated group, and the ERK closure was further enhanced 2 months after ERK punch. In parallel, I adopted an adeno-associated viral vector gene transfer of a constitutive active form of ERK and found that ERK closure was dose-dependent. We observed the newer follicles formation also through this method that could induce a fully ear hole closure upon 2 mm ear patch. Single-cell transcriptomics identified ERK-responsive cellular lineages, indicated in green to yellow, and focusing on fibroblasts, we found that the fibroblasts activating ERK exhibit increased expression of pro-regenerative genes. Looking at the cartilage, we found that the administration of the growth factor mix increased the number of new chondrocytes positive for collagen 2A1, and we could analyze the ECM ultrastructures of the areas of the new cartilage formation through an algorithm developed in our lab. We found the new chondrocytes express both FGF receptor 1 and FGF receptor 3 during regeneration. This study demonstrates that cellular ERK activity controls the delicate balance between regeneration and fibrosis in mammals. And this also poses the basis to identify the limiting factors that impede regeneration in non-regenerating mammals, and to therapeutically target network hubs, such as ERK, to simulate regeneration where damaged organs normally lose their function through scarring.